So we are starting chapter 8, part 2 here with some of those other types of RNA. So we had those three main ones. This is one of them. This is a tRNA molecule, and the main purpose of tRNA is to carry amino acids to the growing chain of amino acids. Now, let's kind of back up for just a quick second. We're going to enter into the second process of gene expression, which is going to be translation. The purpose of this tRNA is going to be to add on the amino acids to the growing chain of amino acids that will eventually become, once it's finished, growing that full chain into a protein. So these guys are going to be carrying amino acids, and as they connect with what is called a codon. A codon is going to be three of those nitrogenous bases in the RNA. They are looking for essentially their complementary three bases, and then they'll drop off that amino acid. So, what is that complementary called? It's called an anticodon. So it's just kind of a molecule that is specific to that codon, so those three bases at a time, in order to drop off the amino acid. If it has that matching anticodon, then the amino acid is going to be dropped off. So it's kind of a weird thing to think about, but this is a helper RNA that is carrying an amino acid. If it finds its complementary three bases in that codon, that anticodon will match, and then it drops off the amino acid, thus adding to the amino acid chain and adding to that to be made protein. So I'm going to have another video kind of explaining how we're going to do this on paper for the exam. So stay tuned for that later on my channel. There are three more types of RNAs that I do want you to know. Again, this chapter is very definition heavy. There are regulatory RNAs, also known as riboswitches, which I think is a neat way to think of them. These are going to be like on-off switches for DNA. They are important regulators of gene expression in bacteria as well as in eukaryotic cells. So I want you to think of them again as on-off switches for DNA. They are going to regulate gene expression. We also have primer RNAs. These are going to be those molecules that we talked about with that animation from before that are going to help prepare kind of the whole situation for the polymerase to come in and start building, right? So we saw this before with DNA replication. This is going to be a very similar process. So these are the primer RNAs. These guys are going to do the same thing. You can kind of think of them as being like the lights on a runway for planes. So kind of a landing strip situation. That essentially, like, a plane can land anywhere, but we want the plane to land in that specific area, and the runway and the lights make it very easy and possible for that to happen. The last is ribozymes. These are going to be like very tiny scissors or, genetic scissors that will cut out extra or known as junk DNA. This will happen in eukaryotic cells only. This does not happen in prokaryotes. Prokaryotes don't bother to cut out the junk DNA in making the RNA and all that good stuff. They just go ahead and go on with it. This does make the eukaryotic process of transcription and translation a lot slower because prokaryotes just don't care. So essentially, ribozymes just cut out this extra DNA that does not code for a protein. To be fair, though, we don't know what junk DNA does, but we call it junk DNA. But it's maybe not the best thing to call it since we don't entirely understand it. All right, we're officially talking about translation. So translation is going to serve the purpose of taking that mRNA, that messenger RNA that we've made from transcription, and then turning it into a protein. I like to get you guys to think of this term translation as literally translating one language into another, like translating Spanish into German or English into Dutch or something like that. You are taking RNA, those A's, U's, C's, and G's, and you are translating it into amino acids. And then that amino acid is going to become what is a protein. You're going to do this using those ribosomes. So the transfer RNA and ribosomal RNA. So those are going to be the quote unquote translators in the process. So we are building a protein using amino acids based on the template of the RNA. So gentle reminder, when we are making that amino acid chain to create the protein, we are using 3 bases or 3 nucleotides at a time, which is going to be called a codon. So those are those 3 A's, U's, C's, and G's at a time. So for example, one codon could be AUG. What this codon is going to be is specifically an amino acid. This is actually one of 20 amino acids that I want you to know. I don't need you to know the other 19. This is the only one that I need you to memorize because it's very special. It is the start codon that is called methionine. You can remember it as MET, but just know that AUG is the codon, methionine is the accompanying amino acid for that codon. Very important. It is the start codon for all of the amino acid chains. It's kind of like the front cover of all proteins. It's the starting place. Kind of like with transcription, we had the promoter site and the termination site. This would be like the promoter site. It's the starting codon, AUG. The termination site is going to be made up of 3 stop codons, also known as nonsense codons. Just know that they're called nonsense codons. You don't need to remember what they are. But that's them. That is how you know that the amino acid chain has come to an end. Now, of these 20 amino acids, there are 64 possible codon combinations that get us those 20 amino acids. We're going to see on the next slide a codon chart. You don't need to memorize the whole chart, but you will need to be able to use them. So remember that AUG codon, and then we're going to practice how to use that codon chart in that other video I said that we would have available here in a bit. So for this codon chart, I do want you to make sure that you know how to use this when it comes to something like A, U, G. What you want to make sure is that you're looking at the positions of the letters, and that is going to help you find it on the chart. So the first letter is going to be this first column, or where we have these first rows. So A is going to be here, so we know that we're going to be looking down this row. Then we're going to come up here for the second letter, and we know that it is U, so it's going to be down this column. And where they overlap is right here. So we're going to be looking in this box, and from that point you can kind of just hunt and find that particular codon, which we see here, AUG, which tells us that it is methionine. You just need to know MET, right? So AUG, that is the codon, how to find it, there it is, and that is the amino acid. Now, I do want to point something out before we move on just yet, and that is that there are a handful of these 64 possible codons that do code for the same amino acid. For example, proline. There are 4 different codons that code for proline. The reason that this is going to happen is it's a safety measure or protection for us or for microbes or whatever organism it is. So there are several codons that will code for the same amino acid, and again, it is a safety net or protection that if replication messes up in that process of transcription or DNA replication, whatever it is, and one of the bases gets messed up and we meant to get CCC and instead we get CCU, then it's not too big of a deal because you still get the same amino acid. Maybe it messes up particularly bad. Instead of CCU, you get CAU, something like that, and you get a completely wrong amino acid, then that could be a particularly big issue. That just depends. We are going to talk about mutations near the end of this chapter, but just know that this is one of the safety measures that is in place with our genetics as well as microbe genetics, in order to make sure that in replication of DNA and in transcription that there's usually not too many problems, not too many issues. All right, I mentioned before that there are some differences between prokaryotes and eukaryotes as far as how they go through replication. There is also differences in how they go through gene expression, so this process of transcription and translation. So for eukaryotes, because of the location of DNA being in the nucleus, transcription must finish before translation starts, which makes sense because transcription is going to occur in the nucleus where the DNA resides, and then once RNA is produced, then it leaves the nucleus and it goes out into the cytoplasm where it then is turned into an amino acid and that protein is produced. Now, when it comes to prokaryotes, it is not restricted by DNA being in the nucleus. So, as soon as the DNA found in the space that the DNA is found in prokaryotes, the nucleoid, the DNA starts going through transcription, starts producing RNA. That little bit of RNA that starts being produced, that product, can immediately start going through translation and start producing amino acid chains and thus that protein at the same time as transcription is happening. So that's fantastic for prokaryotic cells. It's a lot faster. So that's a big difference between prokaryotes and eukaryotes as far as gene expression. In prokaryotes, it's going to be a lot faster. The caveat is it's going to expend a lot of energy. And I mean, it's still going to expend a lot of energy as far as eukaryotic cells, but the process is going to be split up into transcription, stop, translation, and then you have the protein. Meanwhile, in a prokaryote, it's just going to happen kind of simultaneously and expend all the energy all at once, boom, done. But then, once it's finished, that protein has been made. So, again, that big difference, prokaryotes speed up the process of gene expression. Another big difference is that ribozymes are going to only be present in eukaryotic cells, where they are going to cut out that junk DNA within that DNA sequence. So that is going to be those A's, T's, C's, and G's that are not going to code for anything that we can immediately tell what it does. But in prokaryotes, they're not going to cut that information out. They're just going to immediately code that, go through with it. So this is another way that eukaryotic cells get slowed down in the process of DNA replication and gene expression. So in gene expression, we need to have a way to regulate whether or not a protein is being made through transcription and translation or not, because we don't always want a protein to be made. Sometimes those proteins are needed to be made all the time. So what we call that are constitutive. That means that the gene is always on and it is not regulated. Those proteins are constantly being produced. However, when we do need to regulate gene expression, we need to remember terms like repression and induction. Repression is going to be the inhibition of gene expression, specifically transcription. So if you stop the process before it even starts, it's going to prevent the whole thing from being made. A protein will not be produced. So this is kind of the off switch, if you want to think of it that way. And then induction is going to be the on switch, and it allows transcription to even start. So this is that process of gene expression. Now on the next slide, we have a situation on the left without a repressor, meaning that we have gene expression occurring. There is nothing preventing the process of transcription from not happening. On the right, we have a repressor. So remember, repression is that off switch. And in this case, that off switch is a physical roadblock preventing anything from actually going past that point. It is a repressor molecule that is going to stand in the way of the promoter site. So it is a physical thing that prevents transcription from even happening at all. And therefore, that protein will not be made further down the line. Because if transcription doesn't happen, mRNA won't be made, therefore amino acid chains won't be made, therefore the protein won't be made. As an example of where we see regulation of gene expression, I want to talk about the lac operon. The lac operon is a kind of option maker when it comes to the bacteria E. coli. And we all know E. coli, we love E. coli, but what's kind of interesting about this particular bacteria is it has the ability to break down glucose, so that carb that we talked about before, or lactose. So it has options, and it is all due to the lac operon. So again, operon meaning, and I want you to associate it with this, options. So could break down glucose, which I want you to know is substantially easier, and it is E. coli's first choice. But it can break down lactose. It is harder, and it has to go through kind of a process in order to even break down lactose, which is a milk sugar. So There are three important features in the lac operon system. Now, it's kind of a weird thing to think about, but bear with me for a second. All three of these features are all parts of the DNA of the E. coli, and they're actually going in the order that we find it in the DNA. So, what you find first is the regulator, which is a gene that is going to code for the repressor protein. This is going to be the on-off switch for whether or not E. coli is going to be able to digest the glucose or the lactose. So if it's on, it's going to essentially cause the E. coli to digest lactose, and if it's off, that means that it's going to be digesting the glucose. But that's less important, just know that it's the on-off switch. For the control area, it's going to be made up of a promoter and an operator site. So the promoter is the site that's going to be recognized by the RNA polymerase, and the operator is going to be a sequence that acts as an on-off switch for transcription. That third part is going to be a structural area. This is going to be made of three genes that is going to code for, and if they go through transcription and translation, these genes, it's going to make enzymes that are able to break down lactose. So if that regulator allows for the control area to essentially go ahead, is what's going to happen. And there's an image on the next slide. If the regulator allows for the control area to go ahead, then that next step, that third area, is going to go ahead and make those enzymes that allow the E. coli to break down lactose. But again, this is kind of a backup system to if glucose is not available for the E. coli to break down. All right, so here we have a nice image, and what I want you to picture, and I know it's kind of hard because it kind of just looks like a noodle with words on it, but what it's showing us is essentially a strand of DNA, and I want you to imagine this strand of DNA is from the E. coli itself. It's broken up into the three parts that we need for the lac operon system. The first part is the regulator. The regulator, again, is going to create that on-off switch that is either going to allow for the rest of the parts, those three important features, or the two rest important features, to go ahead or not. So it is that on-off switch. If it is on, then that means that it is allowing for lactose to be broken down. Normally, this regulator is going to be off, and therefore it is not going to allow for those enzymes to be made because it does not need to break down lactose unless it has to. So we have the control area that has that promoter and operator, and then after that we have the structural area where we have the genes that are going to make those enzymes that then break down lactose. All right, moving back into something that we have already talked about in previous chapters, and that is the flow of genetic information in bacteria. So remember that this is horizontal gene transfer, and we have three types of horizontal gene transfer that is going to happen. Remember that the goal of horizontal gene transfer is just going to be moving DNA from one cell to the next, and in conjugation, this is going to be through a pilus that is going to move genes from one cell to the recipient, and in the form of a plasmid, to where that genetic information is now shared to the recipient, and it can now pass on that information as well, and that is conjugation. The second is transformation. This is going to be DNA from a dead cell that a live cell is just going to kind of pick up that scrap DNA and then incorporate it into its own DNA. So that is transformation. And then last but not least, we have transduction, where we have a bacteriophage accidentally moving DNA from the original host cell into other cells due to the new phages from that host cell, that original host cell, using the DNA to create those new phages, and then those phages go and infect other cells and then accidentally essentially just kind of drop off that DNA in that process of kind of trying to infect other cells, but they just drop off the DNA. Again, this third one is just purely an accident, completely not on purpose. Now let's talk about something that maybe we've never heard of. These are transposons. Transposons are going to be parts of the genome that can move to other parts of the genome, or move from a chromosome to a plasmid. So remember, our chromosome, as far as our bacteria go, is that one chromosome that is circular, and then moving to a plasmid, which are those little extra pieces of DNA that are also circular, but they are not the chromosome themselves. Sometimes these transposons are called jumping genes, which is kind of interesting and neat. Now, anytime we have seen transposons, about 80% of the time, these are typically unfavorable traits in respect to humans. So something like antibiotic resistance. So we're not sure if bacteria share these transposons on purpose, or if it's just completely by chance that they're sharing this antibiotic resistance, and it just happens to be these particular transposons that they are sharing, sometimes called movable genes. Then here we see that transposon jumping from the chromosome to a plasmid, which is kind of, again, it's a really neat thing because it is a jumping gene or a movable gene. But again, for us, this is usually a gene that allows the bacteria to become antibiotic resistant. So not good for us, but a super neat feature of bacteria. So in bacteria, transposons can actually cause physical changes, like changes to the morphology of the bacteria, like the color could just change, like maybe the morphology would be that the bacteria grew as yellow, but now it grows as green, or something like that. Or it could replace damaged DNA, or again, it can do something really big, really cool, like making the bacteria antibiotic resistant. It just kind of depends, and it has everything to do with the movement of that DNA, that scrambling of the genetics. It can be a good or a bad thing. Usually, it's probably a good thing for the bacteria and a bad thing for us. Or it's a neutral thing for the bacteria, a neutral thing for us. Again, changing the morphological color of the bacteria, not really a good or a bad thing for the bacteria or for us. But becoming resistant to penicillin, kind of terrifying, not a good thing. At least in our opinion. For the bacteria, good for them. Maybe it's natural selection, who knows? Which, speaking of natural selection, that leads us very nicely into this concept of mutations. Mutations are going to be any change in genetic information, so an alteration in the DNA that leads to a change in a protein. So there are three flavors of mutations. They can be beneficial, which leads to essentially every organism on the planet has been a beneficial mutation. There can be neutral mutations, meaning that there is no effect to the cell or organism at all. And then there is, of course, harmful or lethal mutations, which can be very bad for that organism's or cell. Remember that mutations are going to be completely random. It can be very hard to predict, if possible at all, impossible to predict whether or not a mutation is going to be beneficial, neutral, or harmful to that organism. It can also be impossible to predict whether or not there's going to be a mutation that occurs at all and what it's going to be. Mutations are spontaneous changes to the DNA. Now, when it comes to the bacteria that we're growing in lab, it's almost guaranteed that the odds will be in the favor of the thousands of cells that we are growing on a weekly basis, that some of them are going to have a mutation. It's just the luck of the draw and the odds. Now, we would never know what those mutations would be unless we start exposing those cells to different environments and different things, essentially, to test them and see what has changed in them, what mutations have occurred, which, fun fact, we will not be doing because we don't want to test that situation. What we will do as soon as we are done doing our test that we are actually doing is destroy that bacteria, because we do not want those mutations to be passed on, especially if it's something that could cause harm to humans. For nearly all cells, there are two main mutagens, which mutagens are going to be components that make it more likely for a mutation to occur. And these make sense, especially when you think about it. There can be chemical mutagens and radiation-based mutagens. Chemical mutagens can be things like gases, carcinogens, things like that. Radiation is like exposure to sunlight. So it's not a guarantee in the slightest that exposure to mutagens is going to cause a mutation. It's just that it's more likely that it will occur if there is exposure or prolonged exposure to these mutagens. Because remember, mutations are random. It is not a guarantee that they will happen. So just keep that in mind. Remember that a mutation is going to be a change in those bases of the DNA. Now, what we're seeing here is kind of a step further, and it's a change in the RNA. But let's just pretend that we've already seen the change in the DNA. This is now the change in the RNA, and let's just roll with it. It is showing us the change in the RNA, and therefore the change in the amino acid. So the first change we see here, and let me get my little pointer guy, number one, sloppy, is missense mutations. This is going to be a change in just the one base. What this is going to do is potentially change one amino acid. Will this change the entire protein? Maybe. Who knows? Maybe just changing this single base doesn't do anything, because remember, we have that little safety net with those 64 combinations of those codons. Maybe that doesn't change the amino acid. In this case, yes, we are seeing that it does change the amino acid, but maybe in a different mutation, not this one, it doesn't. But in this case, it is. That is a missense. It just changes one amino acid. The next one is going to be a frame shift mutation. Frame shift mutation. So this is where an amino acid, a base is going to either be inserted into this space or taken out. What is going to happen is it causes all of these amino acids to move down or have to move all the way up and over because they have to fill in this space or make space. This is going to cause a massive change in the protein. Very big change. This can be detrimental, a very big problem, very big concern. The third type of mutation is going to be a nonsense mutation. Remember that a nonsense codon is going to be a stop codon, these stop codons. So this is where we have maybe a change in a base that causes a stop codon to suddenly appear before the end of this RNA happens. So it kind of causes this abrupt change, abrupt stop in that RNA, and therefore that abrupt stop in that amino acid chain. So either this protein is unfinished or incomplete, but either way, this can cause a really, really big problem as well. Now, all of that's pretty scary, but really the likelihood of mutations happening is pretty rare. Again, the presence of mutagens make the likeliness of a mutation higher, but it's not a guarantee. Again, mutations are random. It's not a guarantee that it's going to happen if there is a mutagen, but they can happen. Now, bacteria, again, are going to replicate and produce in a very short amount of time and in very high numbers. So the odds of mutations in bacteria is pretty high, and it does happen pretty frequently as far as bacteria go. They're very good at adapting, and a lot of it has to do with their mutations. Mutations are an important part of evolution, and that is something that bacteria have taken a lot of advantage of. It's a very interesting aspect of bacteria in general. We'll talk about that a little bit more when we talk about penicillin in bacteria. Because of their ability to become antibiotic resistant, potentially through mutations as well as through evolution. But mutations are the basis of evolution. So again, the only way that we can really tell if bacteria that we've grown in the lab have become mutated in any way is by changing their environment. But if we keep the environment stable, we'll never know if they've adapted or changed in any way or mutated. But for the most part, we throw them directly into a sterilizer once we finish with them, so we never really know if they've mutated or not, but the odds are very good that they have. And that, my friends, is the end of Unit 2. That is Chapter 6, 7, and 8. Remember that all of that is fair game for Lecture Exam 2. Make sure to be studying. If you have any questions, comments, or concerns, do not hesitate to reach out, to email me. The best way to study, really great way to study, is to review these videos. I am going to be posting another video on Chapter 8, specifically looking at transcription and translation, so in total gene expression and replication, what I will expect of you on the exam, and maybe help give you a little bit of an idea of what you should be studying, what you should do as far as those DNA and RNA sequences. So happy studying, make good choices.